Okay, so we've talked about a lot of organelles within the eukaryotic cell and the prokaryotic cell, the structure of the prokaryotic cell, and both of which are very affected by their cell membrane, their plasma membrane, which we remember is composed mostly of the lipid bilayer. So today we're going to go into a little more detail as to what else is embedded within this lipid bilayer, and then how this lipid bilayer is used to regulate what is transported in and out of the cell. So we'll start with talking about that membrane structure. So we mentioned the phospholipid bilayer, but there's more to it than that. And the model we use to kind of describe the structures within it is called the fluid mosaic model. And it's called that because it's fluid, so it's not necessarily the same static arrangement of molecules. Within the cell membrane, you have these molecules, these compounds, these proteins moving around, and there are a lot of different types. So it's mosaic, it's diverse, and it's fluid, meaning it doesn't stay in one place. So the phospholipid bilayer is made of phospholipids, which have a hydrophilic end and hydrophobic tails, which are pointed towards each other, and makes this bilayer. Then within there, you also have proteins, and these proteins are embedded within there, usually with the nonpolar parts embedded in the nonpolar lipid tails, and then the hydrophobic or hydrophilic parts exposed to the water on both sides. And then we have a molecule called cholesterol. Cholesterol is a lipid, but it also has a hydrophilic part to it as well. And so here you have an example of cholesterol here embedded within the phospholipid bilayer, and it is there for structural support. Okay. So bilayers form spontaneously in water, and the reason is because those hydrophobic tails will start to aggregate towards each other and make these layers. And these phospholipids can then be organized into different functional sections. So you may have, because there's different types of phospholipids. So lipid rafts are made of cholesterol and a type of phospholipid called a sphingolipid. And then these are formed in the endoplasmic reticulum and then sorted in the Golgi apparatus into specific arrangements of lipids, and then embedded within the lipid bilayer. So as we all know, the phospholipids have three parts to them: the phosphate group, the glycerol, and then the two fatty acid chains below them. Okay. The proteins then within there can be categorized into two different types. You can have an integral plasma membrane protein, so that is found embedded within the plasma membrane protein. Okay, you can see all these proteins here. They they are embedded across the span of those phospholipids. Okay, and these are sometimes carrier proteins that carry substances in and out. They can just be channels for water or other substances. They can also be receptors for hormones. And again, their function depends on their shape. The second type are peripheral plasma membrane proteins. Okay? And these are usually used for cell and tissue recognition. So some of them are glycoproteins and glycolipids. So you see we have a glycoprotein here. We have a glycolipid over here. And they can also be shuttle carriers. They can shuttle things in and out of the cell. There is also an interior protein network, so a bunch of proteins which are important to the cytoskeleton. Some of them attach then to these proteins in the lipid bilayer. So they help to maintain the shape, anchor proteins, and help with binding to specific molecules. Now, as we said before, cholesterol is also found within the lipid bilayer. The saturated fatty acids pack together and they make this membrane less fluid. So those van der Waals forces kind of pack them close together. But if you have a cholesterol embedded within there, it's a little more less rigid in shape, so there's a little more space between the molecules. It's not just two tails, so it kind of breaks it up and makes things a little more permeable. So if you don't have enough cholesterol, the lipid bilayer will not be permeable. So some important structural components then. You have this steroid ring structure of cholesterol, you have a non-polar hydrocarbon tail, and then it has an OH group on top, and that OH group is what sticks out and binds with the water because it is polar. The rest of it is non-polar. Okay, so this is an example of structure relating to functions. So you have diverse functions arise from the diverse structures of proteins found within the membrane. And their structural features relate to their role as membrane proteins. If they make, a big hole like that beta pleated sheet we showed before, it will make a good channel. Okay. And the structural components of the proteins, which we talk about when we talk about the amino acids and the different properties they can have, are going to allow the proteins to be embedded within the bilayer, depending on what they are. So there's going to be non-polar portions made of non-polar amino acids with those R groups that can make chemical bonds. You can have transmembrane domains, so large parts of the protein which go through the membrane. They can also help to make pores. Okay. All right. So that's the first part. That's talking about plasma membrane structure. The second part, we're going to talk about permeability. So what is permeability? It is the ability of substances to go in and out or through a specific area. And we're talking about the permeability then of the lipid bilayer. So the lipid bilayer is a semi-permeable membrane, meaning it lets some things through but not all things. And there are two ways in which molecules can pass through the membrane. It can through passive or active transport. And the only difference being passive transport does not require energy, does not require ATP. Active transport does. And we'll break these down into smaller groups or smaller subparts of these. So we're going to start with passive transport. Again, it does not require energy, it does not require ATP, our energy molecule. And in passive transport, if you have a greater concentration of any substance and a lesser concentration of any substance, the molecules will flow from greater to lesser concentration until they are all evened out. So that is the simplest form of this is called diffusion. Osmosis is also something that occurs through a semi-permeable membrane and is usually the movement of water. You also have facilitated diffusion, where you have a carrier protein, and then the fourth one is filtration, or also called bulk flow. So diffusion in a solution where you have a solvent, which would be usually water, and solute, which is whatever is diffused, whichever is dissolved into the water. And if things are diffusing, it will, again, like we said, diffuse from a greater to lesser concentration until they even out. So an example is a drop of dye in water. If you put food coloring in water, it will first start to look concentrated where the dropper is, and then over time it will all spread out to where all of the water has the same color. Osmosis is like diffusion, but through a semi-permeable membrane. So what we have here on this are two solutions with different concentrations of water. Different concentrations of a solvent, of a solute within the solvent of water. And what happens is this membrane is too small, the pores on it are too small to let these molecules through. So in this example, we have a urea molecule. It cannot fit through these pores, this semi-permeable membrane, but the water can fit through. And so to even out the concentration, the water will flow from this greater concentration, from this lesser concentration to the greater concentration until the two concentrations are equal. Okay? So a solution that has the greater concentration of solutes is called hypertonic, and the solution that is lesser concentration of solutes is hypotonic. And then if they are the same, they are isotonic. So water will flow from a hypotonic solution to a hypertonic solution until the solutions are isotonic. Okay? So what happens then is if you have these two solutions with different concentrations of solutes and a semi-permeable membrane, that pressure, or the desire of the water to flow from one solution to the other, is called osmotic pressure. Okay? And the force required to stop that flow can be measured as osmotic pressure. So in a hypotonic solution, a cell will gain water and cause it to swell. So here, if you have a red blood vessel in an isotonic solution, it's going to maintain its shape. If you put it in a hypotonic solution, it will gain water. So water will flow through the membrane and it will start to swell. And if the membrane is strong enough, the cell will reach a counterbalance of osmotic pressure, driving the water in, with hydrostatic pressure driving water out. So this happens with the cell wall of prokaryotes, fungi, plants, and protists. If it's not strong enough, it may burst. Okay, it just can't hold that much water and it will burst. All right, facilitated diffusion again does not require ATP, but provides a way for small molecules that are polar, that can't pass through that hydrophobic layer. It provides a channel then for these to go through. So it requires channels. Again, doesn't require ATP, but they wouldn't go through that semi-permeable membrane without the channel. So these channels can also be controlled by opening and closing them, or they can be selective. So aquaporins are an example of a channel that allows water in or out of a cell. And so water flowing through a aquaporin is going to occur by facilitated diffusion. Now, there are lots of different types of proteins, channel proteins. So ions are these small charged particles, charged ions, charged elements, which are dissolved in solution, and they usually have specific channels they can go through, and they can be gated, where they're open or closed based on this chemical stimulation, or they can just be open. And the flow of ions is determined by the concentration of ions on either side, the charge, so some of them are repelled by negative charge of anions or proteins, or whether or not the channel is open or closed, whether it's gated or not. Carrier proteins then transport ions or other molecules such as glucose in or out of the cell. They bind to the molecule and then it shuttles across the membrane. There is a point at which the molecules, the carriers, are all saturated, meaning they all have whatever substance they are transporting through, they are all fully saturated. They're all bound to it. All right, the fourth one then is filtration or bulk flow. And so this one's a little bit deceiving because there is a requirement of energy, but that energy is upstream to wherever it's taking place. And so filtration or bulk flow occurs because you have a pressure which forces then fluid or a substance through pores due to pressure. So An example of this is blood pressure, which is created by your heart, by the pumping of your heart. That requires ATP. But at the kidneys, you have blood that goes through these very, very small capillaries, and because of blood pressure, it will force out water and waste and ions into the structures of the kidney. All right, our second type of energy or transport is active transport. This requires ATP. So in this type of transport, you have transport from low concentration to high concentration. So this is saying against the concentration gradient, against what is natural to diffusion. And this requires selective carrier proteins. Okay, one type of carrier proteins would be a uniporter, and that moves one molecule at a time from one side to another. Again, it usually requires ATP. Symporters move two molecules at the same time and in the same direction. So here we have a symporter, which uses sodium and glucose. They go through together. And then antiporters would go in opposite direction. So an antiporter would have maybe sodium going through this way and another molecule going through the opposite direction. And these can also be used to describe facilitated diffusion carriers as well. All right, bulk transport then is using membranes to transport substance in or out of the cell. Okay, we talked about vesicles before, how they are formed by endoplasmic reticulum and the Golgi bodies. These can also be formed by just the plasma membrane layer itself. Now, during endocytosis, you have the movement of some substances into the cell. Okay, and there are two specific types. Phagocytosis is going to take in particulate matter, such as maybe glucose or ions, or in this example, you have a bacteria being trapped inside a membrane. And then penocytosis is where it only takes in fluid. Exocytosis then is the movement out of a cell. It also requires energy. And so here's an example of a vesicle filled with some sort of secretion, which then needs to be secreted out. And it has the vesicle, which binds to the plasma membrane, becomes part of the plasma membrane. As it does so, the product secretes out of the cell. All right, lastly, we're going to end with cell signaling. So there's a few terms you need to know. A ligand is a molecule that acts as a signal. So this can be a hormone, such as testosterone or estrogen. It could be another chemical. And then a receptor is what the ligand binds to. And when it binds to it, it'll somehow change the shape of the protein and cause a signal or a change to occur within the cell. These receptors can be extracellular or intracellular, depending on their function. Now, there are different types of signals. So an autocrine signal is one sent to and from the same cell. So it could be from one side of the cell to the other. A paracrine signal is sent from the same tissue. So it would be from one muscle cell to another muscle cell within the same muscle. And then endocrine is a signal sent from one tissue to another. And this would be what the endocrine system is made of, a bunch of organs that send ligands and molecules as messengers to other cells where they are received by their receptors. All right, that's it. Oh, one more. Sorry. Synaptic signaling, which is signaling sent from nervous tissue. Okay, so you have neurotransmitters which can be sent by the endings of a nerve tissue, the endings of a neuron, into a muscle or other type of cell or tissue. All right, now that's it.